Last week we started this chapter 3, and we mentioned the two laws of thermodynamics. Do you remember which is the first law and which is the second law? What is the first law?
Energy conservation.
Energy conservation, yes. Energy cannot be created, it can only be transformed. What is number 2?
I don't think it was in the book.
Yeah, it's in the book. It's in the book.
Oh, is it?
Yeah, it is. Yeah, anyway, there may be things that I'm saying that are not in the book. But this is in the book. So what is the second law? Yeah, the entropy of a system can only increase. So it means that any reactions that happens in your cell, they will only happen if entropy is increased. So if the disorder of the system increases. Okay? So tell me, how is this possible? How is this possible? Because your cell is highly organized. So obviously, your cell is not maximum entropy. It's almost like, cannot find something that is more ordered than your cell, and every reactions that happen inside. So how can a cell always increase its order or maintain it? How is this possible? How can you relate what a cell is with the second law of thermodynamics? What's the explanation there?
Cell releases entropy to outer environment.
Yeah, that's right. So the complete law of thermodynamics is in an isolated system, the entropy can only increase. But a cell is not an isolated system. A cell will always exchange matter and energy with the environment. So when the cell generates order inside itself, it will generate heat. Heat will dissipate in the environment and will increase the entropy of molecules surrounding the cells. Okay, this is actually what happens. So a cell is certainly not an isolated system. Okay? And so this is essentially, what happens, a picture of what I just said. And the first law of thermodynamics is that energy can only be transformed. And the cells are very good at that. The cells are extremely good at transforming energy from various sources and building itself in such a way. And especially one very important reaction, we are not going into details with this, but it's the photosynthetic. Photosynthesis, which is performed by photosynthetic organisms, which are, can you tell me a few photosynthetic organisms? What are photosynthetic organisms? Which organisms perform photosynthesis?
Plants.
Plants, that's one. What else? Plants. What other organisms can perform photosynthesis?
Euglena.
Euglena, yeah, so algae, this kind of thing. Bacteria. Some bacteria can also photosynthesize. But animals, they don't. Okay? So they all rely, all of us animals who cannot photosynthesize, we rely on photosynthesis. Why? Because actually photosynthesis is, it happens in 2 stages. And the first stage is to transform the energy from the sun, the vibration, the energy coming from light, capture this energy and transform it into another type of energy, which we will see later. So this type of energy now is just called an activated carrier. So an activated carrier, it's a small molecule that has captured a type of energy. So during the stage 1 of the photosynthesis, the energy from the sun is captured into activated carriers of energy. And in the stage 2 of the photosynthesis, these carriers will then carry this energy into a different site in the cell, in a different system. This system will actually manufacture sugars and nucleic acid and many, many other things, thanks to the energy brought by these activated carriers. And as you can see, this reaction needs water and generates oxygen, but this reaction needs water and carbon dioxide and generates organic molecules. So this is very important. Ultimately, what our body needs is atoms that are in organic molecules. But we cannot make organic molecules from mineral molecules. That's impossible. Only plants can do that. Only plants, only photosynthetic organisms, they can fix carbon into organic molecules. So if this does not exist, we just starve, because we cannot do that by ourselves, right? So that's very, very important. By the way, what is the most stable form of oxygen currently on Earth, considering our atmosphere? Considering the content of our atmosphere, what is the most stable form of oxygen? CO2. Yes, CO2. So it is not a surprise that CO2 will be the final product of oxidation processes. When we burn our molecules, when we burn our organic molecules, CO2 will very often be the final oxidation product. And why photosynthesis is so important? It's because of this. Cells obtain energy by the oxidation of organic molecules. So this is the photosynthesis. Okay, this is the very general reaction. Carbon dioxide plus water then gives oxygen and sugars. Plants, algae, some bacteria, they use the energy of sunlight. They manufacture sugars and other organic molecules that animals and even plants also use during cellular respiration. So we are burning these sugars. Sugars plus oxygen means oxidation of sugars. Okay, it's just like burning. And this generates water and carbon dioxide. This is performed by most living organisms. And this energy that is generated by the burning of sugars is used to make new chemical bonds, to get energy to build our own molecules. So what you can see is that plants generate oxygen and they take up carbon dioxide. But plants and other living organisms, they will also generate carbon dioxide and use oxygen. So that means that there is a very complex cycle between animals, plants, algae, and bacteria. So animals, during cellular respiration, will generate carbon dioxide in the atmosphere and in water, which will be taken up and fixed by photosynthesis into plants, algae, bacteria. And when they die, they will release this carbon dioxide and everything will be recycled. Okay? So this is a very, very important chain that if this was to be interrupted, we would be in trouble. Okay? And so either the carbon dioxide that plants are using now, they may have been generated by your own respiration yesterday. But of course, as you know, we are generating a lot of carbon dioxide by burning fossil fuels, which is essentially very, very, very old organic material. Okay. that was at some point, many, many millions of years ago, that was actually fixed into organic tissue. So please remember, this is very important. Even though we do not photosynthesize, we rely on photosynthesis for our own metabolism. To understand what really happens, we need to go into details of the chemistry between oxidation and reduction. So as you can see here, oxidation and reduction involves electron transfer. Okay? But what is really oxidation? What does the word oxidation mean? Chemically, yes. Oxidation and reduction means transfer of electrons. But what does the word oxidation mean? Not so precisely, not so chemically. What does the word oxidation mean? The image of oxidation. Just take a look at how it's written and how does it look like.
Add oxygen.
Add oxygen. This is what oxidation is. It's just adding oxygen onto a molecule. But chemically, this is an electron transfer. So let's see a little bit in more details. You consider two atoms, atom 1 and atom 2. And let's say they form a polar covalent bond. Okay? It means that in this covalent bond, one molecule, if it is polar, so one atom will draw most of the electron to itself, and the other atom will have a deficit in electrons. It will be slightly positive, this partial positive charge. So this means that this atom has been oxidized. It has lost electrons. This atom has been reduced. It has gained electrons. So this also happens, of course, in a salt. If you take sodium chloride, which is the reduced, which is the oxidized atom in sodium chloride? Sodium is what? Oxidized or reduced? Chloride is what? Oxidized or reduced?
Sodium is oxidized.
Sodium is oxidized. Sodium, Na+, has lost electron. Chloride has gained the electron given by the sodium. So in an oxidation-reduction reaction, always one is oxidized, the other one is reduced. It's an exchange of electrons. Okay? So the one who is reduced is because something got oxidized. Okay? So always there is two partners in the reaction. This is why it's often called, it's not mentioned in the book I think, but the name oxidation reduction is usually shortened into redox reaction. Reduction, oxidation. So consider these several organic molecules from methane to carbon dioxide. Carbon dioxide is not organic, it's the completely oxidized form of carbon. And the completely reduced form is methane. And when you add one oxygen atom to methane, OH, this becomes methanol, it's oxidized. Then one more bond of carbon is used for another additional covalent bond with oxygen. So this becomes formaldehyde, formic acid, carbon dioxide. So this is progressively oxidized to carbon dioxide. Carbon dioxide is extremely stable, okay, because it's highly oxidized. It has been burned down to carbon dioxide. It's the same thing. If you burn a book, if you burn a piece of wood, essentially the carbon that is in the wood will be released mainly as carbon dioxide. All right. Free energy and catalysis. Chemical reactions proceed in the direction that causes a loss of free energy. Okay, imagine you have a book. Don't do that at all, okay? You have a book. It's made of paper. Paper is mainly cellulose, which is a polymer of carbon. You burn this. You burn this book. What will happen is what? What will happen if you burn this book? Chemically, what will happen? What will happen chemically if you burn this book? Write the equation. Write the chemical equation of this book burning. Paper plus oxygen, yes, paper plus oxygen reacts to give what? Carbon dioxide plus water, carbon dioxide plus water, carbon, yes, very nice. So not all carbon will be transformed as carbon dioxide. So ashes. So this is the thing. But something that you cannot write as an atom or as a molecule in the equation will also be generated. What? Think about burning this. Yes, you will get carbon dioxide, water, ashes. What else? One of the main products.
Light.
Light, yes. Yes, light. But why light? It's heat, right? It's heat. You will generate a lot of heat. So it means a lot of energy that was contained in the atom, in the molecule of the book. A lot of the energy is released as heat. The energy is free. It is freed from the book into the environment. And this is the free energy. Energy is liberated. Now, if I leave my book like this and I come tomorrow, still I will have a book. Okay. This reaction is not spontaneous. Okay. It's not a reaction that will happen tomorrow. For that, I need to do something. What do I need to do to burn the book? Of course, if I leave my book there and come back 2 million years, in 2 million years, this book will have been burned. Okay? So this will happen, but very, 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 very slowly. Okay? But if I want it to happen now, what should I do? If I want to burn this book now, what should I do?
Just fire.
Just use fire. I just have a match. It's nice. I must give a little bit of energy at the start to start the reaction. This reaction will happen, okay? Because the carbon dioxide, the ash, you are increasing the disorder of the book. So this reaction will happen anyway, but it will take a very long time. If I want it to happen now, I must provide a little bit of energy. Okay? And this is the activation energy, so we will see that later. Okay, so think about this free energy that is released when something gets oxidized. Okay? And this action of using this fire, this is actually exactly what enzymes are doing. Enzymes, they are proteins that exist in our cells, and they will catalyze most of the reactions that happen in your cell. And this is what we just talked about with burning the book. Imagine this is the reaction. Y is your book, and X is just ash and carbon dioxide and everything. The energy of the burned book is lower than the energy of the book. So this will happen, but not spontaneously. It needs activation. It needs a small input of energy, and then the reaction will proceed, and the book will burn. Now, using this, if you leave it like this, if I leave the book there, very small fraction of the atoms contained in the book will oxidize, but very little. Very little. Maybe every year, only a few atoms will sort of have enough energy to go and to degrade. So like if you put the book under the sunlight, the book will start to become yellow and will degrade. So this is because sunlight will provide heat. And so therefore, heat will decrease a little bit this activation of energy. And more atoms will become oxidized. Now, if you take an enzyme, it's exactly what it's doing. An enzyme will greatly decrease this activation energy needed for the reaction to proceed. So this is what a catalyst does. A catalyst will decrease the activation energy for the reaction Y to X. Y to X can be many things. Glucose to carbon dioxide and water. Any kind of reaction will happen in such a way. So this is uncatalyzed reaction. This is enzyme catalyzed reaction pathway. So if you now take a look at this graph, the y-axis is the number of molecules in the system. And the x-axis is the amount of energy per molecule. You can see that most of the molecules have kind of a low amount of energy. But some of the molecules have higher amount. They are a little bit more excited. They move about a little bit more. Okay? And without catalyst, without enzyme, without the initial flame, very few atoms, very few molecules will actually get energy high enough to go into the reaction. But now if you add an enzyme, what will happen is that the likelihood of having molecules that have an energy sufficient to undergo this reaction will greatly increase. So this is what happens with an enzyme, right? And if we draw this in a different way, this is a little bit like this, okay? So you imagine a lake that is composed of all these molecules, and they are waves. And some of these molecules are bouncing, but the activation energy is so high that it's very rare when the molecule has enough energy to go onto the dry riverbed and go into the reaction. But when you have a catalyst like an enzyme, what happens is that this activation energy is much lower, so the amount of it will not change the energy of the molecule. The energy of the molecule into the lake will not change. But because this is lower, the energy of this molecule will be just enough to actually go downwards and go into the reaction. Okay? But that's not all. An enzyme, yes, decreases the activation energy, but that's not all. An enzyme also provides specificity. If you imagine this reaction, it's uncatalyzed, and you have this molecule here who is bobbing, alright? And then this molecule can undergo reaction 1, 2, 3, and 4. If there is no catalyst, it's completely random, okay? If these reactions need the same energy, molecule will go to 1, 2, 3, and 4 without difference. But the great thing with catalysts like enzymes is that they will select which reaction will happen. Only one gate, if you want, will be lower. So this molecule will only go to this reaction. And then so, if you have the original molecule, who potentially can give many products, because it can be oxidized in various ways, with enzymes, only one single path will be selected. And this is the kind of things that also happens in our cell, where sometimes what the cell wants to do is to transform this molecule into this one, into this one, into this one, into this one. But imagine the cell suddenly wants to do something, and instead of doing this with the molecule, it wants to do this. What would you do? What would you do if you don't want to do that, but you want to do that with the molecule? What could you do? How the cell could do that? Suddenly the cell doesn't want that, the cell wants this one. This outcome. So what will the cell do?
cells will stop produce an enzyme and produce another enzyme.
For example, so simply if the cell suddenly stops making this very first enzyme and starts making this enzyme and this enzyme and this enzyme, then the fate of the molecule, the fate of the substrate will be different. As with all catalysts, an enzyme will not be destroyed during the reaction. Okay? So you imagine the reaction where you have an enzyme and it has an active site in which the molecule A, the substrate, bind. Then you have this enzyme substrate complex. Catalysis will occur. The product will be the product will be formed and then will be released as well as the enzyme. So the enzyme is ready for the next round of reaction. This is very important. If the enzyme is not regenerated, it's not so very good. Okay? In this case, one enzyme, depending how fast it works, can be used for many, many, many cycles of substrate product reaction. Now, the free energy change for reaction determines whether it can occur. So remember this free energy? Like the energy that is released when you burn the book, all right? Or when you burn a log. What is free energy? You can imagine molecules, they will have different movement. Okay, some molecule will move a little bit, some molecule will move a lot, or will oscillate quite a lot. Same here with, a more obvious molecule. So if you want to see, the molecules of a living cell possess energy because of their vibrations, rotations, and movement through space. Okay? Vibrations, rotations, and movement through space. And because of the energy that is stored in the bond between individual atoms. The free energy, G, in kilocalorie per mole, measures the energy of a molecule which could in principle be used to do useful work at constant temperature as in a living cell. Energy can also be expressed in joules. Okay? Kilocalorie, 1 calorie is 4.184 joules. Okay? So the free energy, this is the energy that is in the molecule, that can potentially be freed to do useful work. Useful work may just be heat. What you want to do is just generating heat. That's great. That's how you do in the winter. But your body doesn't want heat. It wants to use this energy to do something else. Okay? So this is why energy transformation happens. So let's go into more details about this free energy. Imagine the reaction Y to X. If it's energetically favorable reaction, then the free energy of Y is greater than the free energy of X. Therefore, delta G means the difference of free energy between X and Y, is negative, and the disorder of the universe increases during the reaction Y to X. If delta G is negative, Y to X will occur spontaneously. But if it's an energetically unfavorable reaction, the reverse, X to Y, delta G becomes positive. This means the reaction will not be spontaneous. Okay. But I want my cell to synthesize proteins. I want my cells to replicate my DNA. This is not something that can happen if I leave DNA on the bench. It's something that is energetically unfavorable. Synthesized DNA requires a lot of stuff. How can I do that? How can my cell replicate its DNA if it's energetically unfavorable? Burning glucose, yes, fine. That's negative. Delta G is negative. C6H12O6 plus oxygen gives CO2 plus H2O. This is minus 684 kilocalorie per mole. Very negative, very highly negative. It's spontaneous, no problem. It generates a lot of energy. DNA replication? it requires a lot of energy. So how can I do? How can I do to make my key? How can the cell do that?
Cells receive free energy from unstable molecules,
Yes.
or ATP.
Yes. So this is actually what is written here. This reaction can occur only if it is coupled to a second reaction. Okay? So if you have a reaction that has a delta G that is negative, then the energy that is released during that reaction, Y to X, can be used for the unfavorable reaction. Okay? So this coupling is very, very important. Okay, spontaneous reaction, a little bit more words on this one. From the second law of thermodynamics, we know that the disorder of the universe can only increase. Delta G is negative if the disorder of the universe, reaction plus the surrounding, increases. In other words, a chemical reaction that occurs spontaneously must have a negative delta G. The free energy of the products minus the free energy of the reactant must be lower than 0. Then it's a spontaneous reaction. If you take this example here, sucrose is a disaccharide. It's composed of glucose and fructose. And the free energy, the delta G of this reaction is minus 5.5. So this will occur spontaneously. Spontaneously, the sucrose will be transformed into glucose plus fructose. Be careful, again, spontaneous doesn't mean the reaction will occur quickly. Okay. Like the degradation of diamond into graphite is spontaneous because the carbon will actually, go down the energy. But it takes millions of years to happen. So spontaneous doesn't mean it happens quickly. This idea of coupling is this one. So imagine a reaction from C to D which has a negative free energy change. This can actually be coupled to a second reaction that has a positive delta G. So some of the energy used for this reaction will be used for this reaction. So keep this one in mind. Delta G changes as a reaction proceeds toward equilibrium. So this is a little bit more complex. Imagine two molecules, Y and X. The reaction Y to X is favorable. It has a negative delta G. The reaction X to Y is non-favorable. But it happens randomly. At a very, very low rate, it happens. But mostly this reaction will happen, Y to X, because it's favorable. So, for each individual molecule, Y will be transformed into X, and X will be transformed into Y. But the rate of this reaction means that it will occur very often. Eventually, if you take a look 2 hours after you put these together, what happens is that you have very little Y molecules, and you have a lot of X molecules. Of course, because the reaction was energetically favorable. But the problem is, now we have a lot of X, and we have not a lot of Y. So because of this, at equilibrium, so the reaction will come to a stop, because even though this reaction is favorable, there is not a lot of Y molecules available. So and here there is a lot of X molecules, which means that this reaction, even though it's not favorable, because you have a lot of X, the likelihood of X being transformed into Y is higher. So actually, at equilibrium, the amount of Y to X and the amount of X to Y will be the same. And the delta G of the reaction will be 0. So delta G depends on the concentration of y and x. Okay? It depends on the concentration of y and x. So this is what it says here. At equilibrium, there is no net change in the ratio of y to x, and the delta G for both forward and backward reaction is 0. By the way, there is a chemical constant that is called the equilibrium constant, which can be calculated, for example, like this. The concentration of y divided by the concentration of x equals k, or the equilibrium constant. So, for example, in this case, it's 2 divided by 14, it's 0.14. This is the image, okay? It's the image of what the equilibrium constant is. This constant, this equilibrium constant, will be different for each molecule. It depends what molecule you are considering. It depends on the nature of y and the nature of x. Alright, let's go a little bit further. So delta G, this is a different way, again, I mentioned. Changes in free energy occurring in a reaction are denoted by delta G, where delta indicates a difference. Thus, for the reaction A plus B gives C plus D, delta G is the free energy of C plus D minus the free energy of A plus D. So it is also additive. Delta G measures the amount of disorder caused by the reaction, the change in order inside the cell, plus the change in order of the surrounding caused by the heat release. Delta G is useful because it measures how far away from the equilibrium a reaction is. So, this reaction, ATP, is used to make ADP plus phosphate. You can imagine this reaction is negative. The delta G is negative because ATP is the store of energy in our cells. So when you burn ATP, you generate a lot of energy. So this reaction is negative. But the problem is, if you put some amount of ATP in a dish, it will be transforming ADP plus PI, and then slowly it will reach equilibrium, and the reaction will stop. You will not make new ADP plus PI because reaction reach equilibrium, okay? And the amount of ATP and the amount of ADP will never change. What the cell does to make sure that the delta G of this reaction stays negative is, as is written here, it's to continuously make new ATP. So the cell will never let this reaction reach its equilibrium, because otherwise there is no more energy that can be used. Okay? So the cell, by always making new ATP, will ensure that the reaction of oxidation of ATP into ADP plus phosphate is negative. All right? That's a little bit more complex. Right. As I said, delta G can tell you whether a reaction is spontaneous or not, whether a reaction liberates energy or whether a reaction needs energy. But the problem is that it depends on the concentration of the substrate and the concentration of the product. So it's sometimes very difficult to compare two molecules with each other because you don't know what is the concentration. So depending on what concentration and what temperature you are at, you may have very different values of delta G. So we must use another. We must use another concept to compare different reactions together. And this is called the standard free energy change, or delta G0. Okay? This delta G0 makes it possible to compare the energetics of different reactions. Okay, let's see what is delta G0. This is delta G. It is delta G, but it's a little bit, just a little bit different. So for example, now we are starting to use delta G0 here. So this is a spontaneous reaction. As I said, it's not necessarily instantaneous. But for example, this is the reaction of glucose being burned down to carbon dioxide and water. This has a delta G0 of minus 686 kilocalorie per mole. All right? So this tells that this reaction is spontaneous. So delta G0 is very similar to the delta G, but delta G0 does not depend on the concentration of the substrate. Okay. All right, so delta G0, again, you see here. Imagine this is the idea of the coupling using delta G0, and you will see this is exactly the same. So you have the single reaction of glucose plus fructose react to give sucrose. This reaction has a delta G0 of plus 5.5. So you know this reaction is not spontaneous. It will not occur unless you burn ATP into ADP plus phosphate, which will liberate, it has a delta G0 of minus 7.3 kilocalories per mole. So this reaction is favorable. So if you couple both reactions, so delta G0 is simply 5.5 minus 7.3. This is minus 1.8 kilocalorie per mole. So this coupling allows this reaction to proceed because still, thanks to the energy that is liberated by burning ATP, you can actually get a delta G0 that is negative. So delta G0 is useful for this kind of comparison. Again, because it does not depend on the concentration of the substrate and the product. And we can see here the delta G0 for some reaction. Glucose 1-phosphate transformed to glucose 6-phosphate minus 1.7 glucose. So you have a lot of different reactions here. You can see burning glucose is something that frees a lot of energy. So it's very efficient for our cells to burn glucose. All right. The equilibrium constant is directly proportional to delta G0. You remember the equilibrium constant? The equilibrium constant is the relative concentration of product and substrate at which no reaction will be favored. The forward and the backward reaction will happen at exactly the same rate. The chemical equilibria, you can see this one here. A fixed relationship exists between the standard free energy change of a reaction, delta G0, and its equilibrium constant K. For example, the reversible reaction Y to X and the reverse X to Y will proceed until the ratio of concentration X divided by Y is equal to K. The bracket, as you know maybe already, the bracket means the concentration, okay? Moles per liter. At this point, the free energy of the system will have its lowest value. So this is the transformation of Y to X and X to Y, but at equilibrium, this is the point of lowest free energy, and the reaction essentially stops at that point. Okay? There is a certain amount of X, there is a certain amount of Y, but it will not change, and the lowest free energy is reached. And at 37 degrees, we will see how they calculate this later. This is from the book. I will give you more details on how to calculate. They say that at 37 degrees, the delta G0 equals minus 1.42 logarithm base 10 times the equilibrium constant. So the equilibrium constant, if you change this to that, the equilibrium constant is directly proportional to the delta G0. So the delta G0 tells you when the equilibrium constant will be reached. Okay? So for example, for this equation, glucose 1-phosphate into glucose 6-phosphate has a delta G0 of minus 1.74 kilocalorie per mole. Therefore, its equilibrium constant is K equals 10 exponent 1.74 divided by 1.42 equals 70. So the reaction will reach a steady state when glucose 6-phosphate, the concentration of glucose 6-phosphate divided by the concentration of glucose 1-phosphate equals 17. And at this ratio, the reaction will reach equilibrium. Okay? Right. So this is slides that I have added. This is the Nernst equation. Okay? This is the equation that calculates delta G. Okay? And you can see that in this equation, we are using delta G0. So they are definitely linked. So the free energy change in chemistry equals the standard free energy change plus RT times the logarithm of the concentration of X divided by the concentration of Y, okay, for this reaction. Is Y to X favorable? Delta G is negative. Is Y to X unfavorable? Delta G is positive. Okay? I gave you all these lines, I put them online, so you have them, all of them. Okay. For the reaction Y to X, what is really the difference between delta G and delta G0? Since delta G equals delta G0 plus RT times the logarithm of this, delta G depends on the concentration of molecules, this, the temperature, this is the temperature in Kelvin. So you can see when you calculate delta G, the concentration of X and Y is important. When you calculate delta G, the temperature is also important. Right? So this is not so good to use delta G to compare different reactions because it depends on many things. So delta G0, it's a constant that scientists have measured for many chemical reactions. They have measured the reaction at standard conditions. The concentration of the reactant set to 1 molar. The pH set to 7. And the temperature set to 37 degrees. And so, delta G0 depends only on the intrinsic characteristics of the molecules and can be used to compare different reactions. So, scientists have actually measured this free energy change between Y and X at constant condition. So this is why I said that delta G0 does not depend on concentration because it was measured in exactly the same conditions. So the only difference is the difference between the molecules that you are studying. All right. So again, this is what is in the book, but just said in a different way. I already showed that to you. All chemical reactions will proceed towards equilibrium. Okay, so Y to X is favorable until the concentration of X reach a certain point at which Y and X will be in equilibrium and the reaction will not proceed in either way. Delta G will become 0. Delta G0 will still be the same delta G0, but delta G will become 0. But how do we know at what concentration of substrate and product a reaction will reach equilibrium? How do we know what is the concentration of X and Y at equilibrium? How do we calculate that? This is it. All right, so this is the same equation. See, this is for the reaction Y to X. Delta G equals delta G0 plus RT times the logarithm of the concentration of X divided by the concentration of Y. Delta G is in kilocalorie per mole. Mole or calorie? Mole. 1 mole. RT, what is RT? It's the gas constant R, 8.314 joules per mole, and the absolute temperature T in Kelvin, okay, at 37 degrees centigrade, which is 310 Kelvin, RT equals 0.616. Now I want you to explain why I wrote 0.616, because in my calculator, when I do 8.314 times 310, I don't get 0.616. right? I don't get 0.616. So what's happening here? What's happening here? How can you get this RT to be 0.616 while R is 8.314 joule per mole and T is 310? Why 0.616?
We use kilojoule in
Yes,
standard unit.
exactly. So you need to be very careful that this is not only for this course. Most of you are scientists, so this gas constant is very sensitive to the units you are using. So simply in this case, it's not using joules, it's using kilocalories. And then if you transform that into kilocalories, then you will see that this multiplied by 310 is 0.616. Okay. So, at equilibrium, that means that the reaction y to x and its reverse x to y is equal. The same amount will happen. The reaction will proceed in both directions equally, so the delta G is 0. Okay. The delta G is 0. So delta G is 0, which means then you can do this nice equation. If delta G is 0, then delta G0 equals minus 0.616 times the logarithm of the concentration of the product divided by the concentration of the substrate. So then you transform further. X divided by y at equilibrium means that x divided by y becomes k, the equilibrium constant. And then, to make it easier, you transform the logarithm into the log, and then you get, so just times 2.303, and you get delta G0 equals minus 1.42 times the log of the equilibrium constant. And then, the equilibrium constant is 10 to the power of minus delta G0 divided by 1. This is what you have in the book. Okay. This is what you have in the book. I felt like I needed to explain, because they don't really explain why all these figures are different. So just wanted to make sure that you get this one. So, for exactly the same reaction, what does it mean K equals this? It means that for every 1.42 kilocalorie per mole difference in free energy at 37 degrees, the equilibrium constant changes by a factor of 10. So you can take a look here. This is the equilibrium constant in relation to the standard free energy. You see the standard free energy is 0, and then minus 1.4, minus 2.8, 4.3, and so on. And you can see how the equilibrium constant changes. And when the delta G0 becomes positive, then you can see how the equilibrium constant changes. Which means that for every, it's very small, for every 1.4 kilocalorie per mole difference, there is a tenfold difference in the ratio. This is very big, so a very small amount of energy has a very strong impact on the equilibrium constant. Okay, so keep this in mind. All right. In complex reactions, the equilibrium constant includes the concentration of all reactants and products. We already have seen something like this. So far, the equation that I told you, the chemical reactions I told you is y to x. But in this case, is A plus B associates into AB complex. So in this case, you can actually calculate an association rate of K, this is the association constant, times the concentration of A times B. Okay? And then you have the dissociation of AB into A plus B. So this is the dissociation rate, K off, on and off, okay, off times the concentration of the complex. So at equilibrium, the association rate equals the dissociation rate. So K on equals K off. So this is also the equilibrium constant. So it doesn't matter if you have only only one molecule or two molecules, two different, in this case, it's two molecules that react together to give a complex. So that's the same thing. Equilibrium constant is calculated in the same way. The equilibrium constant indicates the strength of molecular interactions. Now, this is very difficult. You will not be marked on that, but this is not a question. They put like this in the book, and they expect you, us, to understand how they calculate this. And this is an exercise you want to do for next week. I will not give you a mark on this one. It's only to see if you can manage. What they say is, consider 1,000 molecules of A and 1,000 molecules of B in the cytosol of a eukaryotic cell. This, they say, the concentration of both will be about 10 to the power of minus 9 molar, right? So 10 to the power of 9 moles per liter. Then they say, if the equilibrium constant K for A plus B, AB is 10 to the power of 10 liters per mole, then at equilibrium there will be 270 A molecules, 270 B molecules, 730 AB complexes. If the equilibrium constant is a little weaker, say 10 to the power of 8 liters per mole, a value that represents a loss of 2.8 kilocalories per mole of binding energy for the sample above, or 2 to 3 fewer hydrogen bonds, then there will be 950 A molecules, 950 B molecules, and 85 AB complexes. So the meaning of this is simply to show you that a small difference of equilibrium constant can have dramatic effect on the binding affinity of A and B. Okay? If you only have 2 or 3 fewer hydrogen bonds, much less complex can form. 2 or 3 hydrogen bonds. This is very small. 2 or 3 is very, very small. But what I want you to try to resolve is how they can calculate this. That is a very interesting problem. That is very interesting. So try to calculate for next week. This is mathematical. This is only mathematical. This is only mathematical. Okay? Of course, you need to, if you know liters per mole and moles per liter, this will make things easier. But this is just a mathematical problem, just to see if you can resolve it. It's very. I spent some time doing this. All right. Do you have any questions so far? It's a little bit chemical, right? It's a little bit chemical. The sequential reactions, the changes in free energy, are additive. We already saw something like this. So if you have the reaction here has a delta G0 of plus 5, and you have this reaction that has a delta G0 of minus 13, it's a little bit different. You can see X is transformed to Y, but in this case the delta G0 is 5. So actually Y to X is spontaneous, and X to Y is not so favorable. Okay? But Y is transformed into Z in a very, very favorable reaction that has a delta G0 of minus 13. So if you want actually to calculate the delta G0 for X to Z, then simply you need to add the two together. And it has a delta G0 of minus 8. So thanks to this reaction, this whole reaction will proceed towards the synthesis of Z. Okay, so it's a simple addition. This concept, thermal motion, allows enzymes to find their substrate. You could say, well, you know, the cells is a bag full of molecules, and these enzymes, you told me that these enzymes can really activate the reactions they catalyze by decreasing the activation energy. Yeah, that's great, but I mean, how can they find their substrate? The cells is extremely concentrated, it has many things inside, you have an enzyme must meet with its substrate. Otherwise, if they never meet, the reaction will never happen. Okay? So how can the enzyme meet the substrate? And you may sort of think why our body is 37 degrees. It's because 37 degrees is actually quite hot. And at 37 degrees, small molecules, typically like substrates of enzymes do not move much. They are big proteins. But the substrate of enzymes, many things, sugars, whatever small molecules, they will move extremely quickly through the cell. And they will move, but in a very chaotic path. They will not move straight like this. They will move in a very chaotic path. They will diffuse through the cell at very high speed and will really make sure that enzymes can always find its substrate at any time because of the concentration of the substrate and because the inside of the cell is always moving because of temperature. Okay? So this random motion of small molecules throughout the cells will ensure that substrate can always find their enzymes. And now this is typical thing that you have inside the cell. I showed you these kind of things before, but you have these ribosomes and protein and stuff like that. You have tRNAs here. These will not diffuse very much in the cytoplasm. This is usually why they are associated with very specific cell areas, like the endoplasmic reticulum or the nucleus or whatever. But small molecules, very freely, they can diffuse through the cell very efficiently. And this will ensure that reactions can occur with minimum delay. All right, what's the time? Now, let's take a look at how we can quantify how fast an enzyme will transform the substrate. And this is a little bit like biochemistry. There are two concepts in how the enzyme functions. Maybe you have seen already, but this is called Vmax, or the maximum speed of the reaction, and Km. Km is called the Michaelis constant, and it's the two scientists who actually did this work who calculated these things. So these two concepts measure enzyme performance. Imagine this graph. This graph is a plot of the substrate concentration in the x-axis and the rate of the reaction in the y-axis. So what you can see is that for increasing substrate concentration, the speed of the reaction increases. But after some time, when the substrate concentration reaches a point, then it tapers off and reaches a plateau that is called Vmax. Vmax is the maximum speed at which the enzyme can work. And Km, actually, Km is simply equals to 1/2 of the Vmax. This is the Michaelis-Menten constant. So you have that in the book. And Vmax is not so useful. Usually we use Km to actually have an indication on how strong, how powerful the enzyme is at transforming the substrate. All right? How can this be calculated? How can this graph be calculated? Well, it is not so difficult. Simply what scientists do is that they take samples in which they know the concentration of substrate is increasing. Okay? The concentration of substrate is increasing. This is very low concentration of substrate, and this is very high concentration of substrate. And then they add to each sample the same amount of enzyme. The same amount of enzyme, say in copy number, in micromolar or whatever. And then, after adding the enzymes, very quickly, because enzymes are very quick. So maybe after 15 seconds or 30 seconds or 1 minute, they will check how much product has been obtained. So this will give a measure of the speed of the reaction. And then they can plot the speed of the reaction they measure, initial rate of substrate consumption in micromole per minute, and then they can plot this reaction. So you can see that at very low concentration, the enzyme will work, but not so fast. But when the substrate increases, then the speed of the enzyme, the speed of the reaction will increase until you reach a point at which the reaction does not increase much, even if you increase the substrate even more. But this is easy here because, well, it's written, but how do you know where is Vmax and where is half Vmax and where is Km? It's very difficult to calculate that from such a graph. So what actually the biochemists they do, it's a very clever bit. They just take the double reciprocal of the graph. And instead of plotting the concentration of substrate and the initial rate of substrate, they actually take the inverse. 1 divided by the concentration of substrate and 1 divided by the speed of the reaction. So what they get in this double reciprocal is that they have a straight line, and then the y-intercept here actually corresponds to Vmax. You just need to calculate back what is the Vmax, and the x-intercept is actually the Km. So this is how actually you can calculate the Km and the Vmax using this kind of graph. Well, anyway, just for the background of this biochemistry concept. Now, many drugs that we take, they are often inhibitors of enzymes. Okay? In this case, for example, you have an enzyme that reacts with a competitive inhibitor that will bind the active site of the enzyme, and this is competitive because it competes with binding of the substrate. The substrate and the competitive inhibitor will bind to the same site. Okay? So if you have a lot of inhibitor, the enzyme cannot function because the substrate will not bind to the enzyme because the competitive inhibitor is there. So let's take a look at what happens to the Michaelis-Menten graph when you add competitive inhibitor. Well, the blue graph here is when you have the substrate only. You can see that the enzyme very quickly will transform the substrate into the product and will reach Vmax. Now, when you add the inhibitor, this is not very obvious here, but when you add the competitive inhibitor, Vmax will not change. So if you go far enough into the concentration of substrate, they will reach the same point. But you will need to add much more substrate to reach the same Vmax. What it means is actually the Km is much bigger. Because you have competitor in the solution, the speed of the enzyme is decreased by this competitor. And if you have, so you have Vmax is about here, that's about half of the Vmax here. And then this is the Km for the reaction without the inhibitor. And this is the Km for the reaction with the inhibitor. So these competitive inhibitors, they actually increase the Km. And you can see this is the double reciprocal. So the Vmax has not changed, as I told you earlier here. The Vmax has not changed, but the Km has changed between these. What do you think would happen to the graph if you don't use a competitive inhibitor, but you use a non-competitive inhibitor? A non-competitive inhibitor is an inhibitor that binds the enzyme at a different site and inactivates the enzyme. So enzymes are very big. They have many different sites. One site will bind the substrate and the reaction will occur. But enzymes have other sites by which they can be regulated. Some of these sites, when you have an inhibitor, the enzyme will just not function. So how would such an inhibitor, a non-competitive inhibitor, what effect would it have on this graph? So keep in mind, it's an inhibitor that does not compete with the binding of the substrate. But when it binds the enzyme is inactivated. What would happen to the graph? What would happen to this graph? It's a little bit more intuitive. If you add a competitive inhibitor, essentially Km increase. So the speed of the reaction is very low. Eventually you reach the same Vmax if you put enough substrate. But what if the inhibitor that you are using is a non-competitive?
I think the result is same as competitive inhibitor.
Yes, but the problem is that when you add a non-competitive inhibitor, it actually works like what I said in the experiments. Scientists add the same amount of enzymes, right? You add to each of the tube the same amount of enzymes. But if in the tube you have non-competitive inhibitor, then what actually happens is that it's as if you were putting less enzyme. Because enzymes will actually, some of them will be occupied by the non-competitive inhibitor. And they will be blocked. So actually, not only the Km will decrease, but also the Vmax will decrease. Because it works like you have much less enzyme. Because some of them will be completely and irreversibly inactivated by the non-competitive. So essentially, it's the same. So Km will change, but the Vmax will also be lower. This is to say that, all about these enzymes, they catalyze reaction. This is very great. They decrease the activation energy for a given reaction, but they will not change the equilibrium. They will never change the equilibrium. You can consider this reaction that is uncatalyzed, something that we have seen before, and few X molecule will be transformed into Y, and a few Y molecule will be transformed into X. So at equilibrium, this will be reached, but this will exactly be the same with an enzyme. Simply, the amount of molecules that are transformed into each other will be higher, but the equilibrium will not change. Enzymes will only change the speed of the reaction, not in which direction it occurs. That being said, some enzymes work only in one way, not the other. Some enzymes can work both ways, so this is not exactly true, but for some enzymes indeed, they work in both directions, and they do not change the equilibrium, even though they catalyze the reaction. Any question? So this is finished with the delta G0, RT, logarithm of concentration of substrate. That's enough. Okay. Do you have any question about that part? No? Okay. Don't hesitate, right? So, earlier in this lecture, I mentioned these activated carriers. So we are going to see what actually they are, these activated carriers, who can capture the energy from one reaction and go somewhere else in the cell and give back the energy to promote other reactions. This is what they do, okay? Essentially, you have a catabolic reaction that is energetically favorable and generates a lot of free energy. It liberates energy that is used to replenish an activated carrier. The activated carrier is there, but doesn't have energy, and it gets loaded with the energy that is freed from this reaction. And then this activated carrier gives back its energy in anabolic reaction to be able to power this energetically unfavorable reaction. Now what I want you to tell me now is, why is these activated carriers needed? Because you don't need that. Imagine if you remove this and you directly have this arrow close to this one. It will work just as well. So you have these two arrows there, and you just put them directly next to each other. You don't have carriers. Why these carriers are important? Can you give me a few reasons of why having carriers that convey the energy, what's the advantage of that? There are more than one, but I'm not sure it's in the book. What is the advantage of having these carriers? We already saw them during the photosynthesis, right? In which the energy of light is harnessed to actually get energy into activated carriers that are then used later to fix carbon into organic molecules. Why do we need activated carriers? Why do we need activated carriers?
The place where catabolism and anabolism occur is different.
Yeah, that's one very important point, is that the place of anabolism and catabolism is very different. So for example, let's say your guts or your liver will generate a lot of ATP that will go to your brain and make sure that you can think. So this is typically, the place is different. What else? What else is important for these carriers? And then I leave you here. So the place, that's one. Maybe there is another one. There is another advantage of having these carriers. Essentially, this is like money, right? This is exactly like having money. Having carriers is like you are selling your cows against bread. You have no money. You are exchanging your cows against bread. If you have money, you sell your cows, and then you can buy bread. This is exactly what these carriers do. So what's the advantage? It's exactly the same. So where is different? If you exchange, you must go to the place where you have somebody with bread, and you must give the cow, and then you must get bread. If you have money, things can happen in different places. What else is an advantage of having carriers?
The energy, it can be used different reaction.
Different reaction. Yes, that's true. Actually, if you only couple these reactions, this is true. That's a third one. So this reaction will only be able to couple this reaction. And if you don't have carriers, like if you don't have carriers that can be used for all reactions, then it's very limited. All reactions need always to be coupled. That's true. That's true. What else? A little bit similar to this one. So if you have carriers, what if I understand what you say is that if you have carriers, then you can use them to power many different reactions, not only one and one. Yes, yes. That's nice. What else? The third one. This is the kind of thing that I do here. It's not only the place, but the time. Yes, the time. You can have these carriers, and at a different time you use them. If you have some possibility for storage, then it's very good, because especially in a plant, these two photosynthesis time, they are different. When different circumstances are present, light versus no light, or when you eat and when you sleep, time is very different. You are doing very different things. So you need to have these carriers for when the time happens where you have no catabolism reaction, and the only thing that you need to do is anabolism. So these carriers enable you not only to separate this reaction in time, but also separate the place where they occur. Okay, so that's enough for today. Thank you very much. So please really try. It's a good exercise. It's not biology. It's mathematics. It's very difficult. It's very, very, very difficult. Well, I think it's difficult. I'm also good in mathematics. It's very difficult. It's very, very difficult. So please try to resolve it next time. So, Emma, you need to start with this kind of thing, right? You need to start with. You need to start with something like that. Where was it? You need to start with coupled reaction. You need to start with something like this. You need to start with this. The concentration of the complex divided by the product of the concentration equals the equilibrium constant. You must use that to resolve the equation. Okay, you can go anytime. Yes. No. Well, you may have one, the final test is maybe about 40 questions, and you may have one or two questions in which you need to calculate. But it's pretty easy. It's pretty easy. Okay, guys. Remind me, what's your major? What is your 何の学部? Science. That's okay. You?
Integrated human science.
Yeah. You may obtain a dispensation for mathematics. What about you? Engineering. So that's a piece of cake. What about you?
Agriculture.
じゃあ大丈夫ね。Agriculture.
No.
But no, essentially no. And if you need to calculate something, it will be. I will not look at your ability to calculate. I will look at your ability to think about the problem, not about the calculation. So, but no, essentially no. It's just for the fun of it doing it.
Last big subject, I was like, oh shit, I understand this. And today I'm like, wow, mathematics. All the equations and stuff. I'm like,
Yeah, I actually did that because I noticed that the ability of students I had previously, I had some Japanese students from mathematics and things like this, and they had a very high mathematics ability. And some of them asked, I don't really understand why in the book they say that, and then this is why I put some details. But essentially, for the book, just read the book. These additional slides that I added is for your information. I will not ask you to calculate something using the Nernst equation without giving you the Nernst equation. So don't worry.
Thank you.
thank you. See you next week.
Thanks, see you next week. See you next week.